Hello students, welcome back to the biology classes. Dear students, in previous lecture we have studied about the gene interactions, and in this lecture we are going to study about the chromosomal theory of inheritance. So dear students, let's start our topic. Dear students, chromosomal theory of inheritance was proposed by Walter Stern and Theodore Bowley. Dear students, as we all know that uh, Mendel gives the law of inheritance, and according to the Mendel, characteristics of an organism are controlled by the unit factor. At the time of Mendel, genes are not discovered. He called gene unit factor. Okay, and after Mendel, Walter Stern and Theodore Bowley in 1902 proposed the chromosomal theory. Uh, according to them, genes are mainly present on chromosomes. Okay, so ultimately a single chromosome may have more than one gene. So those genes which are present on same chromosomes are segregate along with each other. Okay, they are not segregate independent of each other. So the chromosomes which are present on same the genes which are present on same chromosomes cannot show segregation and cannot show independent assortment. These genes are known as linked genes, and we are going to discuss this topic in our next lecture, the linkage. Okay, and here we are going to discuss some features of the chromosomal theory of inheritance. So dear students, there are some important features of chromosomal theory of inheritance. The first is as the sperm and egg cells serve as the connecting link from one generation to the other, all hereditary characters must be carried in them. Okay, so we can say that the gametes, actually sperm and egg are gametes. Gametes transfer hereditary characters from parents to the offspring. They are the connecting link. Okay, the hereditary factors are mainly present in the nucleus of cell. We have already discussed this topic, this point in the previous lectures. And like Mendelian alleles, chromosomes are also found in pairs. Okay, homologous chromosomes, we have already discussed the homologous chromosomes. The homologous chromosomes are the chromosomes which have same number of gene, type of gene, or position of gene. Okay, they are known as homologous chromosomes. So we can say that like genes which are present in pair. Okay, the paired genes are known as allele, and the chromosomes are also present in pairs. Okay, these chromosomes carry gene. Okay, there are large number of genes present on chromosomes. Okay, we can give an example. The chromosome number one of human beings nearly has 3000 genes. Okay, approximately 3000 genes are present on chromosome number one, whereas the least number of genes are present on chromosome Y of humans. Okay, that has nearly 231. Okay, so there are large number of genes present on the chromosome. Those genes which are present on same chromosome. Okay, so or we can say that the homologous chromosome, they cannot show segregation, they cannot show independent assortment. Okay, only the chromosome show segregation. The genes which are present on same chromosome, they are known as linked gene. Okay, and this is known as linkage. Okay, they cannot show segregation. And next is the sperm and egg have haploid set of chromosomes. Okay, after fusion they form zygote, and in the zygote is diploid. So after fusion, when zygote is formed, then diploid stage is re-established. Okay, and homologous chromosomes form synapse during meiosis, and during the formation of synapse. Okay, in 11th class, when you studied meiosis. Okay, in the meiosis you have studied prophase 1. In prophase 1 there are 5 stages. I think you all remember that: leptotene, zygotene, pectine, diplotin, diakinesis. Okay, if you don't remember that, so please see that lecture. Okay. In the meiosis, what we have see that in prophase 1, the homologous chromosomes came in close contact with each other. Okay. When they came in close contact with each other, there is an exchange of segments between non-sister chromatins of a homologous chromosome, of course, there. So that is known as crossing over. Okay, that is known as crossing over. Or due to the crossing over, the linked gene may show segregation or independent assortment. Okay, that is known as incomplete linkage. Oh, don't worry, we are going to discuss these topics further in our next lecture in detail. And these are the main features of the chromosomal theory of inheritance. Actually, the segregation and independent assortment is shown by the chromosome mainly. Okay, there are some exceptions. Okay, the crossing over, due to the crossing over, the linked gene may show segregation, but normally the linked genes show very less segregation. So here we can say that segregation mainly occurs in the chromosome. Okay, and you can simply see the diagram. And what you have seen that during the cell division, or at the time of cell division, or we can say that at the time of gamete formation. uh, Actually, in 11th class you have studied about the meiosis. Okay, actually meiosis occurs in the germ cells, okay, which are mainly responsible for the formation of gametes, sperm and ovum. What you have studied that in S phase the DNA replication occur. Okay, after DNA replication, the chromosomes segregate. Okay, the chromosome segregate uh, at the time of gamete formation, at the time of, and each gamete have haploid set of chromosome, haploid set of chromosome. You can simply see in the diagram. So according to the chromosomal theory of inheritance, the independent assortment is also shown by the chromosomes. The genes which are present on the same chromosomes cannot show independent assortment. Okay, they show linkage. Okay, uh, so the homolog actually the segregation and the independent assortment mainly occurs in the chromosome. Okay, not in the gene. Okay, genes which are present on same chromosome, or you can say that the homologous chromosome, cannot show segregation. Okay, at the time of gametogenesis. Okay, here the segregation and independent assortment is mainly shown by the chromosome. Dear students, initially the chromosomal theory of inheritance is very controversial, but in 1930 it is supported by the Alnor, and after that in 1950 T.H. Morgan worked on the fruit fly and proposed the chromosomal theory of linkage, and his work supported the chromosomal theory of inheritance. Dear students, there are some similarities between gene and chromosomes. Okay, that is known as parallelism between the gene and chromosome. The first is both genes and chromosomes help in transmission of genetic information from parents to the offspring. Okay, and similarly, genes control any trait, and as genes are present in the chromosome, chromosomes also control the traits or characteristics of an organism. Okay, similarly, both genes and chromosomes have specific number. Okay, and both gene and chromosomes replicate during the cell cycle. Actually, they replicate in the S phase of the cell cycle. Okay, similarly, the genes and chromosomes uh, segregate genes which are present on the homologous chromosome. okay, not on the same chromosome, segregate at the time of gametogenesis, and they are transferred to the gametes, and actually when they pass they form haploid cells or haploid gametes. See, similarly, at the time of fertilization, when gametes fuse, the diploid state is restored. Okay, both genes and chromosomes again came into the diploid form, or we can say that the in pairs. Okay, similarly, chromosomes and genes also show segregation. Okay, uh, the genes which are present on different chromosomes they show segregation, but the genes which are present on same chromosome cannot show segregation. Remember this thing. Okay, and also they show independent assortment. Both gene and chromosome show independent assortment, but remember one point, that the genes which are present on same chromosome, they cannot show segregation, they cannot show independent assortment, so they are known as linked gene. But the gene which are present on different chromosome, they show segregation and independent assortment. So this is the parallelism between the gene and chromosome. uh, Dear students, now we are going to talk about the chromosomal theory of linkage. Actually, we have already discussed that Morgan experiment on the fruit fly, that is known as Drosophila melanogaster, and he proposed the chromosomal theory of inheritance. Uh, there sometimes a question is asked that why the Mendel selected the fruit fly, or what are the advantages of selecting fruit fly as a experimental material. So the main advantages of selecting Drosophila melanogaster as the experimental material is that it is easily available. Okay, it can be reared inside the bottles in the synthetic medium. Okay, and their generation time is very short. Okay, they can be reared in within the 2 weeks. Okay, uh, the generation time is very short, only 2 weeks. Okay, after 2 weeks they start producing new individuals, and similarly, and we can easily distinguish between the male and female Drosophila based on their body size. Okay, males are larger size. Oh, sorry, the male are of smaller size as compared to the female Drosophila. Okay, and they have 4 pairs of chromosomes. Okay, which are of different size, and we can easily extract the chromosomes from their salivary gland. Okay, and we can easily breed them. These are the main points. Okay, so on the basis of his experiment, Mendel proposed certain main important points. That first is he discovered the basis of variations. Okay, uh, he discovered linkage and distinguished linked and unlinked genes. He also proposed chromosomal theory of linkage. Okay, he proposed chiasma type of hypothesis, showing that chiasma cause crossing over. Okay, and he also found the frequency of recombination between two linked genes is directly proportional to the distance between the two, and he also worked on sex linked inheritance, and he gives the theory of inheritance. So dear students, the sex linked inheritance and linkage, okay, and the crossing over, we are going to discuss these topics in our next lecture. So I hope you all understand. Thank you.